So on behalf of my colleagues at the National Eye Institute, I'd like to thank the sponsors and the organizers for the opportunity to speak with you about our work on AREDS. Part of my talk will be on AREDS too, but I'll spend the majority of my presentation discussing the role of omega-3 fatty acids and their putative impact on factors and processes associated with AMD. So we'll start with the AREDS. I'm not sure if members of the audience can relate to this experience that these schoolboys are having, but this is an old photo of a school nurse administering some cod liver oil. And so I had the opportunity to speak at the University of Wisconsin a while back to a group of people who were diagnosed with AMD, and many of the people in the audience were able to relate to this. So we'll be speaking a bit about the lipids that are present in this bottle that these boys are enjoying so much. I'd like to start with a reflection. I won't be speaking that much about dietary macular xanthophylls, but I just thought it would. I've always been impressed by this passage from Steinbeck, and he speaks about energy in plants. And I first came into the field at a Gordon conference where there were many botanists, and so this is something about resource allocation in plants. And he writes in The Grapes of Wrath, the sun flared down on the growing corn day after day until a line of brown spread along the edge of each green bayonet. The weeds grew darker green to protect themselves, and they did not spread anymore. And this is about photopigments and protection and energy, and how all these factors can sort of come together to promote survival of a certain of this plant. But I think that there are similarities between some statements that Richard and John have made in the past, and I found that quite interesting. And the fact that this wasn't discussed at the conference, I thought it would be valuable just to mention it. But they write that in the advanced form of AMD known as geographic atrophy, the foveal center, which contains the highest concentrations of lutein and zeaxanthin, tends to be spared until late in the course of the disease. And sometimes what we see is this horseshoe-shaped ring, and it's sort of the same idea. We have this concentration of photosensitive pigment that acts in a protective capacity, and I just wanted to sort of point that out because I didn't hear anyone really speaking about this issue. And so to move forward, I wanted to just present another quote, again from Richard and John, and I think it's something that will set up my talk. But basically, back in 1997, they made the observation that understanding the mechanism of macular pigment accumulation and the transformation was really essential. And so what my talk will mainly be about today is regulatory mechanisms and metabolic fate, and how we can take information from epidemiologic studies and apply it in this capacity. So what I'll be speaking about is moving from these epidemiologic studies to some mechanistic studies, and then back to the human population. So that will be basically presented in the context of some unifying concepts, and that's that we have to really understand the enzymes, transporters, ligands, receptors impacting or impacted by the nutrients we're studying. And so I'll speak about how uptake, transport, retention, and repair. We've talked about these things in some capacity over the last day and a half, but how they may be impacting the disease we study in macular degeneration, and how they can be studied in these larger genome-wide studies. So I'll start with the AREDS. We have a website, and anything that passes in conversation, you can certainly glean from our website, but I wanted to put this up. AREDS 2 is a phase 3 clinical trial. It's a controlled trial on 4,000 people. We're administering 10 milligrams of lutein and 2 zeaxanthin per day, along with a gram of omega-3 fatty acids. There are 4,000 people, approximately 4,000 enrolled, and this was the original plan. So there's a first line of randomization with the nutrients up here with the omega-3s, and with the xanthophylls we have another arm that is a combination of both. And then there's a second tier in which we administer the AREDS type supplements, and these include beta-carotene, zinc, and variations on the original formulation. So we are studying the response to these drugs or these nutrients within 82 centers across the United States. And I just thought I'd say something. The way that this is different than AREDS 1 is that the people that we're enrolling this time around are at moderate to high risk of macular degeneration. So they come in with bilateral drusen, or they come in with bilateral drusen in one eye and advanced AMD in the other eye. In this case, the study is the eye with a bilateral drusen. The eye at risk is the one that has not progressed to AMD. And so our primary outcomes are progression to advanced AMD. So this is a structural outcome. So we're looking at central geographic atrophy and atrophic form of macular degeneration in which the RPE and photoreceptors degenerate. We're also looking at the wet form of choroidal neovascularization, where there's this florid pathogenic form of angiogenesis that takes place in advanced disease and puts us at risk for sight loss. So our secondary outcomes include functional outcomes, vision loss. This is best corrected visual acuity, so we're testing this annually. And then we also have measures of lens opacity and some outcomes in cardiovascular health and cognitive function as well. So all of these are part of the study, and it's a 5-year trial. As I said, we're about 3 years into it, and we've enrolled around 4,200 participants, and this is basically how they pan out. We have about 1,000 people in each arm of the arms for the primary randomization, and then about 3/4 of the people have elected to go into the secondary randomization with the AREDS type supplement. And again, we have the original formulation on which we published in 2001. We have a low beta carotene group for smokers, low zinc group, and then we have a combination of this novel formulation. So, just some findings that we have. We enrolled about 60% of the people that we enrolled were in the bilateral drusen category. So these people are at moderate risk. About 1/3 have advanced AMD in one eye. The age at enrollment, most of the people were 65. So the way that you read this is, as you go around the horn, these are the youngest people, and they come around here. So most of the people were 65 or over, with about 4 in 10 being between 65 and 75. 1 in 4 people were between 75 and 80, and then about 1/5 of the people were greater than 80 years. And so this is just a quick summary of some demographic data that we have. And the way that you can read this graph is that these are proportions. So slightly over half of our participants were female. Almost all were white. 7% were current smokers. 1 in 5 people had a diagnosis of existing cardiovascular disease. Approximately half take aspirin regularly. 50% report use of statins. It's an educated population. 4 in 10 people have a college degree, and then 90% are on multivitamin supplement that we're providing as part of the study. So our closeout is in December 2012, and I just wanted to also announce that we'll be starting a genetic study. We've just recently been funded for that, and that should allow us to continue on some of the work that I'm about to present. Okay, so I'd like to switch gears, and we'll speak about the role of omega-3 fatty acids now in the retina. And so the issue of plausibility in studying any nutrient-disease relationship has certain key features, and I think that everything I say in this slide can be applied to what we know about xanthophylls. So basically, it's very important to look at independent, intake-dependent and modifiable nutrients that actually need to be consumed to change the status in the retina. And in the case of the macular xanthophylls, John spoke yesterday about the fact that we cannot biosynthesize these de novo. It's the same issue essentially with the omega-3s. We need the parent fatty acids in order to synthesize these. So they're also concentrated in the retina, as macular xanthophylls are. They're localized to certain areas in the retina that are susceptible to the pathology that we study. Biophysical and biochemical capacity of these nutrients to act on factors and processes that are associated with AMD. This is something that I'll speak about in the forthcoming slides. But again, we've spoken about this stuff in with the macular xanthophylls. And so the slides that follow will be basically dealing with this issue. And then the fourth concept is that we need some demonstration in large human studies to sort of support this as a reasonable idea. And so I'll, as I move through the talk, I'll deal with these four concepts. So I talked about this issue of enzymes, transporters, ligands. I think this is where I've been focusing my work. And I think this is certainly, as we move forward with these large-scale genetic studies, we should be able to study these issues in greater depth. So, okay, so what are the putative actions of these omega-3s? So I'll speak mainly about icosapentaenoic acid, which I'll refer to as EPA, and docosahexaenoic acid, which I'll refer to as DHA. And I'll speak about their role as ligands to nuclear hormone receptors affecting transcription. I'll speak about their role as precursors to certain lipid mediators, and I'll focus on three of these concepts. I'll show you some animal work that we've done, but we'll start by just moving forward through the evidence base. So we talked about the fact that we need some support from human studies, and we learned in 1995, Julie Morris actually was the first author on this study about the relationship between dietary intake of these lipids and the decreased likelihood of macular degeneration. And since that time, there have been approximately 15 other studies that have resulted in produced results of similar magnitude and direction. So if we look at advanced AMD, and we look in these continents across these varied populations, we see very similar results. And essentially, there's a decrease in likelihood of AMD of approximately 70% to 40% among people that report high intake of these lipids. And it's the concordance of evidence is actually quite strong when you look at advanced AMD. And so as time goes on, there are more studies that are basically coming out. These are observational studies, so our inference can't be as strong as it would be from a clinical trial. But again, the fact that these results are clustering together gives us a very good reason to move ahead, and they were the basis for moving forward on AREDS. At the time that these studies were presented, there were also a number of mechanistic studies that came out, and I'll speak about them. Specifically, there are metabolites of omega-3s known as neuroprotectins, and they're quite potent. They're potent autocoid paracrine hormones that act in the retina, and I'll speak a little bit about that. We have conducted some studies in mice on a retinal angiogenesis, a feeding model, in which we simulate the diet that humans have within animals and challenge them with an oxidative stress model. And then recently we have a paper out in Science on another compound known as PPAR-G, to which this DHA lipid binds, and I'll speak a little bit about that. Okay, so if we start with the first study, it's a feeding study in mice. We used a feeding paradigm and a transgenic mouse that concentrates DHA and EPA. Our phenotype was essentially a neovascular phenotype. The vascular bed is slightly different than the one that we see in AMD, but you know, our opinion is that this is sort of a general process, and since at the time we did this research there was not any, essentially any good model for neovascular AMD. We went with this one, and basically the take-home message is that we saw that neuroprotectin D1, these resolvins that I'll speak about more, and this PPAR pathway are quite important in reducing the likelihood of having this angiogenic, pathologic angiogenic response in the retina after exposure to lipids in the diet and through this transgenic animal model. So we published this in 2007, and basically I can speak a bit about the endpoint, but it's a hypoxia model. It's an oxygen-induced retinopathy, and where we expose the animals in infancy to high oxygen tension and high oxygen, and then we return them to room air, and there's this florid outpouring of growth factors that leads to this proliferative vascularization. But so this is a section of a normal retina, this cartoon, and what we find is something called vasoobliteration. So the vessels drop out, and then there are these tufts that actually leak protein and can cause inflammation. So this is what we'd expect to see in a wild-type mouse raised in room air, and this is the result of our oxygen exposure. So what we did was we introduced a diet, as I said, and I can show some of the results of this diet. So the VO represents vasoobliteration. This is the dropout of vessels. There's an omega-6 diet, which is more like the US diet, and there's this omega-3 diet in which we administered 2% DHA and EPA, and you can compare across these images here to see that, you know, there's vascular dropout here, and this one looks relatively normal, like a wild type. There was about a 40% reduction in the area of vascular dropout in the omega-3 group. And so this is our transgenic system. This mouse overexpresses DHA, and we find exactly the same thing. So the take-home message is that exposure to these lipids is in the presence of this hypoxic insult is protective. So that's the case of the vascular obliteration, the dropout of vessels. So now let's look at the neovascular tufts. Again, we see a reduction within the dietary model among the area of the vascular tufts, and we see the same thing in the transgenic mouse. So, you know, at this point, it was important. I spoke a little bit about the receptors, the enzymes, all these factors that may act on the omega-3s or on which the omega-3s may act. And so we were very interested in these neuroprotectins. And they protect against cell death. And the lipids that I discussed actually act as the precursors to these particular molecules. So we were very interested in the expression of these neuroprotectins within the mouse after exposure to these insults. And so, as it turns out, we were able to measure them only in the omega-3 group. And once we did that, we quantified the volume of these neuroprotectins. We injected them back into the eye, and we exposed the mouse to oxygen. And again, these were the three that we looked at. They were on that diagram that I showed. They were the resolvins and this neuroprotectin. And again, we see about a 20% reduction in the area of this vasoobliteration, in this area in which we find the dropout of the nascent vasculature. In the case of the neovascular area, this florid, these tufts that I spoke about, the effect wasn't as large, but it was significant. And so we felt that that gave us a basis to move forward. We recently published a second article on another factor. We use the same feeding paradigm in this case with double knockout mice, and there are 4 major enzymes that work on the omega-3 fatty acids within the eye to produce factors that are capable of modulating vascular function. And so we took a look at each of these factors. These mice were fed with omega-3s. The white bars represent the wild-type mice, and the black bars represent the double knockout. So these mice don't have functioning enzymes. So we feed the lipids within these two groups. Okay, within this cyclooxygenase group, there was no difference, which meant that the omega-3s were able to rescue the mice without the COX-2 gene. They were able to rescue the mice without this cyclooxygenase 2 gene. It was the same for this lipoxygenase gene, but in the case of this 5-lipoxygenase gene, it was clearly a difference, and the omega-3s were not able to rescue the mice from this insult. So, as it turns out, they need this particular enzyme in the case of the exposure. So they're using the omega-3s as a substrate to produce either a metabolite or something that protects the mice from this oxygen. So this is the LOX5. So I'm going to move this up to the upper corner here, and if we ask what the LOX5 actually does, is it produces these hydroxylated DHAs. And the question is, what is the bioactivity in these DHAs, and do they exist in the human? Because we're going to move now from the animal to the human. And so this white bar represents the concentration of this compound known as 4-hydroxylated DHA. The black bar represents 7. And basically, there's a hydroxylation on this 4-carbon or the 7-carbon. And so we went back to the mouse and we looked in the retina. So this is in human serum. So what we find in human serum is the 4-hydroxy-DHA. What exists in the mouse retina? Because we need to look in primary tissue. And it was the same sort of pattern. So what we decided at this point was that it was important to look at this particular compound and what it actually did. So I spoke a little bit about these peroxisome proliferator activated receptors. They're very important in gene transcription. And what we found was that if we administered this 4-hydroxy-DHA, the message, the mRNA for this PPAR receptor was actually upregulated. So the next question is, what does that really have to do with the vascular pathology? So we blocked the PPAR, we blocked the PPAR receptor with a drug, and that's what exists on the left side of this diagram. Okay, we blocked it, and then we put the animal in the oxygen, and we were interested to see what happened. This is an animal that was administered a saline vehicle, and this is an animal that was administered the drug, but also the 4-DHA. And what we find is that there was, again, a reduction in the amount in the area of neovascular pathology. So this basically tells us, so a number of things, that DHA is actually the precursor to a potent metabolite that is processed by this lipoxygenase enzyme and affects the peroxisome proliferator activator to actually inhibit the sprouting and proliferation of new vessels. So if we go back to those human studies, this is one mechanism that may be acting in the system. So we may be able to target this PPAR gene with the combined therapy of DHA to actually affect the system. So this is a link between the human studies and the animal studies. And as I showed, we also were able to block the PPAR with certain drugs. So this is the concepts again. So with that, I'd like to move into some human research that we've done on some large-scale human studies. So some large-scale human studies that we've tested in the age-related eye disease study. So the take-home message on this is that we were able to use a systems approach to identify a certain pathway, the AKT-PI3 kinase pathway, and use it to develop certain classification models in macular degeneration. We're able to look at people's genes, the variation in people's genes among elements in this pathway, and actually classify people accurately post hoc, just with the genetic information alone, and that will be the message at the end. So what I did was we looked in the animals again to see which genes were affected by diet, which genes were dysregulated with exposure to the omega-3s. And so we had primary tissue from the mouse, and we had also a human retina that was grown in culture. It was a differentiated retina. And so we fed the mice as I described in the past. And in this cell culture model, we we added DHA for 14 days, and then we looked at the dysregulation of genes. And it ended up to be around 240 in the mouse and around 1,400 in the human. The reason why these are different is that the chip that we used to analyze the results was a different chip. But as it turns out, there were about 100 genes in the mouse that were dysregulated by the omega-3s. There were around 500 in the human. And so what we wanted to know was whether these genes were also associated with AMD. So we expose the mouse or the retina, the model retina, to omega-3s. We look at the dysregulation in the genes, and then we see if these same genes are associated with AMD. So what this diagram represents, these are p-values from the studies on our gene study. So we did multivariable tests on the relationship between SNPs and AMD. The red symbols represent the genes from there was 500 genes from the human model, so we looked for variations, SNPs, within those genes that were dysregulated by omega-3s. We did the same thing on the animals, and those are the blue symbols. And as it turns out, if we looked at the nature of this variation in the genome, there were quite a few genes, SNPs, variants that were present in areas that affect regulation of protein synthesis or gene expression. And so I can just move ahead, I can just skip. But the point here is that we found some interesting results. So what we did from this point, so this is the diagram that I showed a couple slides ago. Anything in this yellow box is significant at a p-value of 0.005. And so we took these associations with AMD, and we went into a bioinformatic resource, and we tried to filter them. We tried to discover how they may act within certain pathways with some software that we had. And as it turns out, I'll just give an example. We found that they were enriched in this synaptic long-term potentiation pathway. So I'm just going to warn you that the next slide is a bit busy, but it's just a demonstration of how we actually can move from these genome-wide results into a pathway. And so this is a pathway. It's hard to see, but the point is what we're looking for here is genes that were dysregulated by exposure to the omega-3s and also associated with AMD and also acted as drug targets. And so we found 4, and we were very quickly able to go from our animal studies into to discover these genes, which have not been previously associated with AMD, but this one, the protein kinase C, or some of the protein kinases, have been associated with proliferative diabetic retinopathy. So these are membrane receptors. This is in the glutamate pathway, but we're able to actually target these genes now within our animal model. So at this point, we would go back to the animal model and look to see whether using these drugs actually had an impact as well. So we went, again, from this expression study to the SNP AMD relationship to drug targets. We also find relationships among genes that are not targets of drugs, and so this is a very enriched pathway. Anything with a color on it was associated with AMD. So this is basically how we go through the process. So another way that we do this is to look at an agnostic procedure where we have a curated lipidome. We have a set of genes that have been associated through some proteomic methods with interaction networks and signaling networks. So we start there, and these genes are related to lipids, energy storage, cell membrane structure. These are all lipid-related genes, and we go into the genome at this point. There are 23,000 protein-coding genes in the genome. There are 3 billion base pairs. And so within our lipidome, there were around 2,200 genes. Every red bar represents a gene. These are chromosomes, these are genes. Okay, and so what we're going to do is we're going into a chromosome, and we were able to do this with our genome-wide association study. So this is chromosome 10. We're going to focus on a single gene down here, which is a plextrin homology gene. So we've expanded this. These are the protein coding regions, anything with a dark. Within the gene, we have SNPs, and this is actually our unit of analysis. So we go from the genome. We go from the genome to a chromosome, which is the red bar, to a gene, to the SNPs. And basically, we use different gene chips. This is a plextrin homology gene. And we compare the allelic frequency the alleles within our sequence variant. And so you can be homozygous, you can be heterozygous, or you can be homozygous dominant. And so this is just an example. In the plextrin gene, we control for age and smoking in our analysis. And so we look at what's known as the minor allele frequency. In this case, let's say it's A. So the likelihood of having an A, it's 18% in the people without AMD, it's 32% in the people with. And so this is basically the way that we compare these SNPs across the genome. We have 11,000 SNPs. In this particular analysis, these are our candidate SNPs. These are SNPs that were significant with AMD and associated with lipids at 0.05. Our candidates, as I said, and again, we went to the bioinformatic resources, and we found that these SNPs loaded in the AKT-PI3 kinase pathway. Now, I'll explain why that's important in a second, but basically, there are studies. Now, how does this link back to the lipids? And I guess this is the key point. We have to come back once we do this genome-wide scan and make some sense of this. And there are papers that have discussed the role of DHA as a positive modulator of AKT. This is a paper that appeared in JBC about 5 years before we even did this study. And so it's just a way that we're able to link this information together. And it's a mechanistic study that shows that DHA increases this phosphatidyl serine, which is a membrane phospholipid concentrated in the retina, and this affects these plextrin homology domains, which affects AKT translocation and PI3 kinase activation, which is involved in angiogenesis. So this was a way that we went from this genome-wide scan into a plausible mechanism, and we can take this back to the mice and then identify influence points within the AKT-PI3 kinase pathway and actually drug those areas and see how they may be affected. So this is just a follow-up. I just wanted to, I think just for lack of time, we came up with 19 SNPs, and these are the chromosomes on which they existed. And so we plotted these SNPs. We have to interpret things in the context of known loci. So what we end up doing, there are established genetic risk factors, and you'll see that 2 of our SNPs were resident in genes near the CFH, this complement factor H gene, and in this complement factor 3, C5, and this 10q26 area. And so what we had to do was remove these SNPs and redo our analysis. I'll come back to this in a second. But basically, after we did that, this was our p-value. It was 10 to the minus 27. We were able to predict who had AMD from these 19 SNPs, or these 9 SNPs when we took the others out, with a reasonable degree of certainty. About 8 out of 10 people that had AMD actually were predicted to have AMD with our models. And so when we ran the statistics, it was, as I said, about 10 to the minus 30, 10 to the minus 20. And so it's just a method that I think I wanted to share with you, something that I think has great promise. When we do this AREDS2 study, we'll have more genetic information. I've done this on the macular xanthophylls as well. It's not in the same advanced state as this is, but I just thought it would be sort of valuable to speak with everyone about this. But we end up doing something called a permutation test. So everything I showed until this point was based upon parametric statistics. And we can also go back and do a more stringent test. And this is an example of a permutation test in which we scramble the phenotype and the genotype, and we replicate this hundreds of thousands of times, and we get a p-value and develop something called a null distribution. It's what we would expect by chance alone, but we build the distribution ourselves. So this is actually an exact p-value. And so again, we were up about 10 to the minus 27. So this is a pretty strong result. And again, it sort of opens this venue in the AKT-PI3 kinase pathway. So I've basically talked about this, and I think, so I can probably end now. So the general message is that, as I showed in the human studies that were started back with Julie's first study in 1995, people reporting higher intake of fish and nutrients concentrated, this should be omega-3 fatty acids, we're about half as likely, 20% to 50% less likely to have macular degeneration, advanced macular degeneration than their peers. So then, so if we look at the genome-wide association data, I'm pretty convinced that we can efficiently go into this and make some very reasonable inferences about what may be happening. And I showed you the 2 mouse studies, and that led us to the conclusion about the resolvins, the neuroprotectins, and the PPAR-G, and we're working on this right now. There are follow-up studies that we're doing, and then this is a metabolite of DHA that we're also working on. So we were able to go from these epi studies into the mouse, and hopefully we'll be able to go back to the human. So I just have basically reiterated this point, and I think we can skip this and just go on to the acknowledgement. So I worked with a large group at the NIH, the National Institute of Alcohol Awareness and Abuse, with Norman Salem's group. I worked with Lois Smith's group at Harvard. At the NEI, I'm working in the Division of Epidemiology and Clinical Research. And then the human work was contributed from my colleagues at the Retina Foundation of the Southwest, and Ricardo Wowie, who's actually at University College London now. So with that, I will end. So thank you for your attention.